Hello and welcome to today's Sesnon Speak Up series. Our event today is called Interpreting Life with Viruses, Humans and Plants with Amy Youngs and Russ Corbett-Detig. And this is part of a virtual exhibition and interactive website called What Makes Us Human? An Art and Genomics Convergence, which we launched in November of 2020, and it's up forever. But please check us out at whatmakesushuman.org. My name is Shelby Graham, and I'm the director of the Mary Porter Sesnon Art Gallery here at UC Santa Cruz. And we are honored to host this event. Also, we're sponsored by the UC Santa Cruz Arts Division, the Genomics Institute, and Open Lab Collaborative, at UC Santa Cruz. And I'd like to thank our Sesnon Gallery manager, Louise Leong, who is organizing this whole event and giving us technical support. So thank you, Louise. Today, we'll be looking at the concept of ways of translating art and science. And this conversation, as all of our Sesnon Speak Up conversations, aims to create a safe space to talk about critical issues of our time. And this is certainly a critical topic that we've been discussing about life, interpreting life with viruses. And we've all been interpreting more data than ever. So our specialists, our experts today are going to teach us how to interpret and visualize this data. Let's see. I do want to just mention that the program is being recorded and it will be available in a couple days on our website, as well as the Sesnon Gallery YouTube channel. And please check out our Sesnon YouTube channel for all of our recorded events. And you may ask questions anytime using the chat function, and we'll incorporate them into the conversation and also at the end during our Q&A. Before we begin, I'd like to offer our land acknowledgement, because most of us are here at UC Santa Cruz in the Santa Cruz area. And UC Santa Cruz is located on unceded territory of the Awaswas-speaking Yupi tribe. The Amamutsan tribal band, comprised of the descendants of the indigenous people taken to Mission Santa Cruz and San Juan Bautista during Spanish colonization of the Central Coast, is today working hard to restore traditional stewardship practices on these lands and heal from historical trauma. So please find your local land acknowledgement, and I welcome everyone from all across the country. I know there's many of you tuning in from Ohio today, and hopefully around the world. We'll be discussing, again, teaching and learning in the arts and sciences, and explore how to participate with data, and you're in for a visual treat. And here to introduce this complex project is UCSC Professor of Art, Director of the Open Lab projects here on campus, and the curator of What Makes Us Human, Jennifer Parker. And she will introduce all the speakers today, along with our moderator, Isabel Bjork, who's the executive director of the Genomics Institute. So please welcome Jennifer Parker. Thank you, Jennifer.
Thank you, Shelby. I'm so delighted to be here today, and I'm really thrilled to be able to share this project with you. What Makes Us Human is a virtual exhibition that I designed as part of the Arts and Genomics Initiative that we initiated in 2019. The exhibition itself is just really driven by curiosity and intrigue and sort of the general desire to expand research and think together collectively across the arts and the sciences with technology and humanists, really wanting to spearhead new ways of working together that sort of de-silos us. In some ways, I think of the archive of this exhibition, the index, as a kind of library that you can surf and move around in. The reason why these projects are visualized as spheres is because there's really more to it than these squares that we've been living in. You know, this idea that there's something hidden, something on the other side is hopefully intriguing enough for you to want to click on it and learn more. So please visit whatmakesushuman.org. But with that, I'm really excited to engage with our panelists today. And we have Amy Youngs, who's an associate professor from Ohio State, and Russ Corbett-Detig, who is a biomolecular engineer and affiliated with the Genomics Institute here as well. So with that, I'm just going to hand it over to Isabel Bjork, who will be our panelist host for today. Thank you.
Hi, everybody. Thank you so much for coming. Our line of operation for today really is having 5 minutes presentation from each of our fabulous experts here, from Russ and Amy, and then a series of discussions around the interconnections of their pieces, what it means in this world. What I wanted to contextualize for everybody is to let you know that we started the Arts and Genomics Initiative because the Genomics Institute is a cross-divisional, multi-institutional organization that really looks at genomics research from a number of angles. And one of the things that has been fascinating is the impact of visualizations generally on the work that we do. And so Jennifer and I came together some time ago to talk about what does that mean and how do people respond to them. And so that's why we're here today, is really to think that there are these links between what we do professionally in the small sense of science and the bigger picture of how people react to it, how they interpret it. And it has been a fascinating experience seeing it through the lens of art and the professionals in the art world. And I'm very grateful to all the artists here that have put this together. Thank you. And that's specifically to Jennifer and Shelby and Louise and Amy. So with that, we would like to begin with Amy, who is giving a 5-minute overview of her work, and then we'll go to Russ. I encourage everybody on this call to put all the questions that you have in the chat. You can do it now as they speak. You can do it later. We will have a series of questions and answers after their short talks. Thank you. Amy?
Thanks so much. Thanks for inviting me. I'm always so fascinated with these projects that are, I like Jennifer's word, de-siloing. It's really great. I always learn a lot. So I'll just introduce myself a little bit and talk about my work. I'm an artist, and I'm working to expand the ways that we understand and engage and interact with non-humans. And I have a practice of living with plants and worms, and this has changed the way that I consume and make art, and I think about who I am in the ecosystem. So for me, data information is actually gathered through close attention and embodied relationships. And as an artist, my challenge is really to try to figure out how to share that with others. I'm thinking about it as humble data here. The fact that humans are totally reliant upon and continuous with a multi-species world is clear and very scientifically known, yet completely ignored by Western industrialized societies in the way that we live. So we know that we need to have a major adjustment that's required for the survival of our species, but how do we start? And how do we recognize that our vulnerability, that we are vulnerable and that we need to shift our focus towards respectful relationships with our non-human kin? So this long-term relationship that I've had with the colony of composting worms has really provided me with a regular reminder of the importance of waste transformation. So this worm bed webcam is a broadcast project called Live Feedings. I was hoping to share with others an appreciation of these invisible laborers. So normally hidden from view while they're doing their work in the dark, worms, pill bugs, mites, microbes, they're all, they're here, and some of them are made visible here through infrared camera technology, which doesn't harm them. In this handshake with composting worms, I wanted to feel vulnerable and to recognize myself as being a part of an ecosystem that includes worms, mites, bacteria, insects, people, cameras, internet, soil. A handshake is a ritual of greeting, an acknowledgement of equality, a sealing of an agreement, and demonstration of respect. So I held the worms in my hands, and I felt the gentle movement of their fleshy, moist bodies fed by the waste generated from my household. This worm colony has been with me for over 25 years. So living with these worms, it always reminds me of the blurry boundaries between food and waste, dirty, clean, decay, and renewal. And it reminds me that my human life isn't really possible without the work that they're doing and associated with their entangled symbiotic communities. So this time-lapse color-enhanced footage allows human viewers a way to experience this worm data in a somewhat unrealistic, but at least visible way. With hubris, humans, we sometimes imagine that our machines can replace living things. And we wonder, like, what would it be like if there was a machine that could turn waste into something useful? And this ideal machine wouldn't use gas, electricity, or batteries, and its byproducts could power the growth of something nice like flowers. And actually, that's what worms already do. These red wiggler worms turn household waste into fertilizer. And this is a project called the Machine for Living Interdependently, which is an interface that's designed to connect a colony of composting worms to humans within a domestic space. So the worms here are offering us in-home waste disposal services. They eat our leftover newspaper, tea bags, veggie scraps, and then they generate inline fertilizer for the healthy growth of indoor plants. So rather than spending our time collecting and separating the worm compost, you just pretty much participate by rocking in a chair, which is attached to a siphon bulb that pushes the water up to the top of the ecosystem. And the water percolates through the system, becoming infused with the worm-generated fertilizer, which is then directly feeding the plant roots. So partnering with these living ecosystems benefits humans. Plants are providing the cleansed air, the worms are keeping the waste out of the landfill, and this project is really meant to share this very known information about interdependence, but in a way that's at our human scale, so somewhat more approachable than just kind of imagining it all happening underground. My work is also really inspired by science. So for many years, I've been co-teaching an art and science class with a molecular geneticist at The Ohio State University. So crossing disciplines like this is a real exercise in humility because we're total amateurs in each other's fields. But it also allows us to both become a little bit more open to the feeling of not knowing while teaching. Iris Meyer is my collaborator, and her research involves the symbiotic relationships that fungi and plants share. Mycorrhizal fungi must find plant hosts in order to survive, and the plants have encouraged this symbiotic relationship for millennia. When fungi symbiotically team up with plants to trade nutrients, a dance has to occur that includes a kind of molecular trust. So the fungi are invited directly into the plant roots to trade minerals for sugars. So here you can see the nucleus is helping the fungi enter. And scientists really want to know more about this relationship. Like, how do the partners recognize each other? How do they establish both open channels and boundaries? There's a lot to learn here. As artists, though, I mean, we really struggled to relate to this abstraction. I mean, the video is really helpful, but it's like, we can't really see or feel any of this. So we tried to figure out ways to gain an understanding of this topic of underground symbiosis. So we practiced scientific methods like experiment design, microscopy, thin layer chromatography, and then we kept field notebooks to document our scientific experiments and our artistic experiments. We did drawings and ideas. And then really in the end, we were super inspired by this Scientific American blog post that says, the world's largest mining operation is run by fungi. So this new knowledge really puts humans in our place. It reminds us that we are not the most industrious. Scraping minerals out of rocks, fungi deliver them to plants. So for our project, we cast the human participants as fungi who would bring the minerals into a fungal tunnel. These are the paper bracelets that served as models for the minerals that the fungi mine. So this installation, titled Where Rocks Are Fed to Trees, sought to make science tangible and to go beyond visualization. An embodied participatory experience was created that engaged all of the senses. So we were modeling scientific facts, but that was brought into a human scale and a sensory understanding through movement, sound, vision, smell, and taste. At the end of the tunnel was a rock candy treat that participants exchanged for their mineral bracelet. And it included a short text which said, You're a fungal bodied particle on its way into the depths of a plant's private parts, its roots. Delivering your mineral, you're rewarded with sugar. Use it well, grow your network, and trade in peace. So the experience allowed humans to enact, in a sense, the story of underground symbiosis. And what we hoped is that it offered them a way to kind of connect with this incredible complex relationship between non-humans, which is hidden from view. And we were hoping that that would offer a sense of wonder and humility for humans. Thank you.
Thank you. That was fascinating and beautiful. I can't wait to unpack that a little bit. So now we have another lens through which we see science and art, ultimately, which is offered by Russ here, who will talk about the genomic side of the work that is happening.
Sure. There we go. Hi. So thank you, Isabel, for introducing me. And Amy, that was fascinating. I have questions for you later. And first, I'll just introduce myself. So I'm Russ Corbett. I'm a professor of biomolecular engineering. And until this last year, I pretty much exclusively studied evolutionary genomics. And I would say that my work is widely read in a very small community. And in this last year, I've pivoted quite a lot, as have many people, to try to contribute my efforts to work on COVID and studying SARS-CoV-2 genomes. And it's been an interesting event because it sort of catapulted the kinds of things we do where we may previously have made really complex, really information-dense figures that are interpretable only to an incredibly small subset of people, into making visualizations of these data that are supposed to be evocative and interesting and informative to anybody, regardless of their background and how much they knew coming into it. And that's, I hesitate to ever call myself an artist, but it has been a really interesting journey to figure out how to convey this information in useful ways that anyone can consume, hopefully. And I'm going to show you just a few images that my team and others have produced as a part of this effort, and then show you a really kind of amazing interactive framework for exploring some of these data that I find just sort of beautiful and simultaneously interesting. So anyway, follow along. Hopefully this will work. Cool, that's not it. My apologies. Let's try this one more time. Okay, how about that? Nope, still the wrong one. Okay. We'll just do, I need 1 second, having technical difficulties. So we'll do that. Okay, one more time. This is the one. That's the right place. Okay, sorry about that, folks. You know, we're 1 year into this pandemic and my life is basically governed by Zoom and I still struggle with it almost every day. And that is one of the most exhausting things I experience. And I apologize for making you all suffer through that event with me, and hopefully it wasn't too terrible. Okay, so what I'll talk a little bit today is about trees. And I mean this in the context of how we understand the relationships among genomes. And I'll explain that as I go, so not real trees. These are sort of metaphorical trees that we use as a way to understand these things. Before I get into that, I'm just going to show you a few of the images that other groups have made as a part of this as well. So this is an image produced by a team called NextStrain, and what it's meant to show you is all the ways that we can tell that the virus has gotten around the world during the pandemic. So you see in cases over here, we know that it arose in China, almost certainly, and then transmitted out. So each of these links is tracing one of those inferred transmission events, right? We say, okay, we know that a genome from South Africa is related to one from China, so we know there was an event that drags these together here. And this is a way just to show, I think, if you lean back, you kind of see that this is a really amazingly spread event that has gotten kind of everywhere in the world from everywhere else in the world. And we're this incredibly interconnected maze of transmission of the virus that's come out of this sort of stuff. And of course, you know, this is sort of a summary of that information that I was talking about earlier, and I think is a really powerful way to see that, because you can immediately see that, okay, everything coming to Iceland seems to be coming from Europe as far as we can tell, and similar kinds of events are immediately apparent just by staring at these data and how you can understand how countries are linked. Another figure that we've made in this is in a lot of our work now is exploring how to use color to just immediately tell you something useful. In this case, we're trying to show you the evolutionary neighborhood. So you may have a new genome and you want to know what it's related to. And the warmer colors basically correspond to places that we think it's more likely to live, the neighborhood it might be in, where red would be, okay, here's a group that looks just like this. And as you go further and further out into blue and gray, we're sure that that isn't what we're seeing here. And it's a way to think about the uncertainty in this information and a way to just sort of encapsulate what is hard to understand by just displaying a giant list of numbers. You can see in this color just very quickly, okay, there's a spot that I think this goes to. And that's what I'm trying to get out of this and try to display. And then finally, this is one of those trees I was talking about, right? And so what we're showing you when we do these kinds of things is links between samples that we infer to be related to each other. That is, they share a set of mutations. That means they must come from some common ancestor. And when two samples are close together, they tend to be very, very close relatives. And when they're really far apart, like this light blue one to this sort of dark purple over here, they're pretty distantly related to each other. They've had many, many events separated from each other. And these can be a really useful way to display this kind of information because it tells us things very quickly. And as I said earlier, it can tell us about the sort of evolutionary neighborhood that you're in. So for example, this cluster here is what you've heard called the UK variant, B.1.1.7. That's what's going on with this long thing here. And you can see it's kind of special, right? It's super distant from the middle. It's got its own expanded framework. And so without even knowing the core underlying reasons that we know it's special and interesting, the idea is these images can tell me that very quickly. It's sort of blasting off the page to the lower right here. And with that, I'm also going to go over to show you kind of how we explore these trees when we're using interactive frameworks. So this is, again, this is a tree our team produces using our software, but we're displaying it in that NextStrain framework I talked about. I really encourage people to go check out that site and try this themselves. You can do exactly what I'm about to do here, or not exactly, but something very similar. And the really great thing about this is that it not only displays all that same information, right? These are the same sorts of neighborhoods. Out here is that special UK variant in the same colors, but it's also explorable, right? So it's interactive and I can really mess with it. So for example, if I want to zoom in a big way and just see everything closely related in this section, we can zoom right down to it by clicking on this branch. And we see the sample names, we see the dates they were collected, all sorts of information is displayed immediately to make us follow along and understand what's going on in that little neighborhood of the tree, right? So I can drill down very quickly and zoom out. And the other thing I love about this that I find just sort of beautiful and wonderful is that there's often different useful ways to show the exact same information. And one cool thing about this framework is it allows us to do that. So earlier I showed you what's called a radial tree. So you can reorient the thing interactively to show me that same information. This is the exact same tree structure, but we've moved around where the samples are located such that you can follow them in sort of different ways. And the same is true again with unrooted, which I like for all sorts of things where, again, the exact same information. Here's that special cluster B.1.1.7 out here, but it's got a different way of seeing it. And this can be, I think, this visualization in particular is a really good way to see just how special that one is, how far out it is, and how unusual it seems to be relative to basically everything else we know about so far. And that's kind of the gist of this stuff. And so again, the site I recommend you check out is NextStrain, and they'll have all kinds of cool versions of this. They track flu, they track Zika virus, all sorts of fun stuff. And yeah, it's been a great journey. I'm excited to talk to you guys about it. Thanks.
Thank you, Russ. Everybody, I want you to know too that after this, we can send out some links that have been referenced during this talk as well, so that you don't have to scramble right now with all the things that are in chat and have been discussed. So to start, I want to take a moment to acknowledge how difficult this year has been and thank everybody for coming here today. Our work has been so affected by COVID, clearly. And it has changed how we interact with each other, with our areas of expertise. Russ, you just talked about how your portfolio of work really has pivoted, right? Because now you are so focused on the virus. I'm wondering if you can talk a little bit about the reshaping and reimagining that's occurred. You talked a little bit about that in the context of having, reaching out to new people. And if you could sort of elaborate on that with regard to what it means for some of these visualizations you just showed.
Sure, yeah. the biggest group of people we're trying to reach right now is public health officers, right? So there's a transformation that's about to happen because in large part driven by the pandemic, where we've realized the value of genome sequencing of pathogens and what that information has. And so what we're doing right now in a big way is trying to figure out how to make this information consumable to a person who may be trained in epidemiology, but doesn't understand the genome science necessarily, or may not be trained in it. And make it really, really straightforward to explore these things in very intuitive ways. And that's something that's sort of the exact opposite of how science typically works, where we make incredibly complex, jargon-dense things that no one else understands to try to switch this around to something that I can immediately see and understand what that's trying to tell me. And of course, this will be valuable beyond public health. I think the general public will start to use these kinds of tools and interpret the same sort of information that public health officers have, but at least in the short term, while the pandemic is ongoing, I think our target will be the people that have the biggest chance of making an impact on health outcomes.
Thank you. And Amy, I'm wondering if you can speak about that too. You have interconnectedness as a core aspect of all of your work. Has that been affected by this last year as well?
Yeah, I guess so. I've often been really interested in this space of communicating online, and I've done projects that are webcam oriented, and thinking about the interesting ways in which that opens up spaces for artists that are less hierarchical. And like I can now travel to conferences without having to be there or get on a plane. And colleagues that I have in other countries can be here even potentially. And I like that non-hierarchical sense of communication can happen in these times. I guess I'm always looking for the silver lining a little bit here, despite our feeling quite lonely, that we do have ways in which we're able to take care of each other, even emotionally through these networked spaces. And I'm also really heartened to see how the students that I work with are understanding that they also have agency to figure out how to do things together without the teacher, and care for each other and share with each other in ways that don't really require, let's say, authorities. So I'm interested in that networked ability.
Thank you. One of the things that struck me really early on about your work in particular, and then also the language around Russ's work, is that trees, the idea of trees, are central to the terminology that's used. And we've talked about branches, we've talked about leaves, we've talked about, we've seen the plants and even the earthworms. So there's central images and themes in both these categories of work. And on my mind recently has been Richard Powers's 2018 Pulitzer Prize winning book, Overstory. And so there's a quote in there that I wanted to share with everybody and get your reactions to in terms of how it relates to your work. So it's, a forest knows things. They wire themselves up underground. There are brains down there. Ones our own brains aren't shaped to see. Root plasticity, solving problems and making decisions. Fungal synapses. What else do you want to call it? Link enough trees together and a forest grows aware. So maybe, Amy, we could start with you. Can you tell us if this statement resonates with you and your work?
Yeah, definitely. I've been super interested in networks, and also loved reading that book among a host of other really interesting books that are in a way challenging our hierarchy of humans, but also of intelligence. The idea that we have to have a brain to be intelligent is something that I think that quote challenges. There's so much intelligence in the non-human world that's been here so much longer than we have. And there's so much that we're learning from them. And that's partly, I think, why I'm very interested in this word humility, because I feel like we actually have to take that position, a different position, when we're trying to understand the non-human world. We actually have to go, oh, actually brains and intelligence might not necessarily be the only way to have intelligence. Maybe we need to look at it from a different perspective, in a sense. So I feel like that's been really a useful way to approach learning and thinking about things from new perspectives.
Thank you. And then Russ, it struck me when you were speaking that there's that last piece of the Powers quote, you know, the link enough trees together and a forest grows aware. I'm wondering, you know, your thoughts around stimulating awareness through these images.
Yeah, sure. Well, I think one of the key messages of the whole thing is that we as humans are all in this together, right? This is a global event that is occurring in a very real way through all of our populations. And I think it's pretty easy to see that when you look at these kinds of trees. And in fact, if you drill down into them, you'll see that for every sequence or every genome that we see from California, as often as not, its closest relative is from some other country. It's getting around, it's between all of us. And I think that has been a really important component of communicating this information to people, is that it isn't just your neighbor, it's everyone. We're all sharing this all the time. The other thing I think is worth touching on is to go back to your quote, and a lot of Amy's work seems to be about symbiosis. That is essentially how organisms interact with each other. And I think there's a really interesting parallel to the virus, which is certainly not a beneficial interaction. Amy, most of what Amy's talking about are things that are probably good for both parties. In the case of the virus, you know, it's good for SARS-CoV-2. It isn't good for us, but there's this incredible interplay where our behaviors are shaping the virus itself, right? When we do a lockdown and we don't communicate, we suppress it. We're pushing this population down. And of course, it's, you know, indirectly, it's not consciously choosing this probably, but it is indirectly causing us to make those sorts of decisions. So it's also shaping us simultaneously. And these kinds of coevolutionary processes are incredibly fascinating on this kind of time scale. And I think it's a really interesting connection that's what fascinates you. And I think it's sort of central to what I'm talking about right now.
Thank you. And then I think that the corollary to that is this focus that both of you have on education. And so you're both experts in your fields in science and art respectively. And then you're also both professors, and you've dedicated your work, you know, not to the private sector, but to education. And so I'm wondering if you could speak a little bit about that idea of interconnection and being able, the evolutionary responses and the matrix that develops with how you're explaining your material to students and why it matters in their lives. Amy, maybe we could start with you, because you talked about some of the going into the imagery itself and experiencing it.
Yeah, I think I always have a really hard time actually explaining why it's important to students. I think I fail at that every time. So I think that's where it became really important to have the students go through the process with me. You know, I don't really understand this stuff. I'm not a scientist, but I really am curious and I want to get to understand things better. Let's see what are the ways in which we can understand things together. And I think that having them as part of a community of engaging new ideas and new ways to maybe even tell stories to each other about new science that's happening, like how do we grapple with this together and how do we come to terms with it and share it amongst our small community of teachers and students. So I guess for me, it's really just been more of a symbiosis with the students rather than, you know, one in which I'm telling them that it's important. We make it important together is how I think that it works best for me.
Yeah. So I'll say that I agree with this. I've both learned from my students and hopefully they're learning from me, at least at some rate. And one of the coolest things we did, I'll go into detail. One of the coolest things I got to do this year was teach all of our first year graduate students about how to use these tools and methods. So I had them mimic a public health officer. Here's your first 10 genomes. Tell me what happened. And the amount that I learned from watching them struggle through tools that we've tried to make intuitive and easy was incredible. And I have learned that I need to be better at this, of course. And so we're working all the time to improve these things and to help automate the interpretation process. So definitely learned a lot from them. With respect to what I think I'm teaching them or hope to be teaching them, as I said, what's going to happen in the next generation here is that we're going to reinvent the way we do public health. And genomics is going to be central to that. And I think, the more that we train these scientists early and get them on board to understand this information, they're going to get to be the people that do the real innovation, right? They're the ones starting grad school now. This is the time for them to take over this process and do this. And so I'm really just delighted to be able to start to show them the kinds of things we're thinking about and to get their ideas about how to make it better. And so that's been, I guess, an incredible symbiosis as well of knowledge exchange with the students. And I've absolutely loved it.
Thank you. Well, a reminder to everybody, please feel, free and do send questions through the chat. There is a comment/question here that I wanted to link to what Russ was just talking about. So it says, are these static graphs? And can you interrogate the data? So that's some of what you were just talking about, Russ, and Amy too, in terms of touching and feeling it, but maybe you could explore that a little bit more, in particular, the value of having the static imagery and what you can use it for, and the value of having interactive and process-oriented images as well, that you can look at it through a time scale. Let's start there because there's a series of other questions connected to that. It might get too complicated. So let's go for it.
Cool. Do you mind if I start, Amy, or you can? But okay, sure. So yeah, the static images are incredibly valuable when you need to set something down that is just meant to be displayed exactly this one way. And that's often the case in a scientific paper, and I think probably quite a lot of books are like this as well, where this is the thing you're meant to see, exactly this and nothing else. The interactive tools are obviously, I think, a more powerful way to get intuition for this sort of information, because you get to see how it's constructed. You get to drill down into little details if you want to. And I think that can be maybe more useful ways to onboard people to understand these things, because they get to take them apart. And then, how the other value of interactive tools is that depth of information, right? So you can show that zoomed out view that's just colored points and connections, but when you zoom in, you see sample names, you see the country it's collected in, the date it was collected, all sorts of what we call metadata that gives me the context for that specific dot I put in this image. And I think that's one of the really valuable things about these sorts of tools, is that it can be one framework where I see multiple levels of information and I'm able to go into those little edges and then all the way back out as I need to.
I have to say, I was totally in love with that moment where in your data set, you moved it from the circular round data into the flat graph, and it just completely helped me understand how those two things were connected in a way that I couldn't get before. I did not link them together in that way until I saw that animation. So it seems really important to have all of it, because that's what worked for me, and I think that I actually needed the still images and then the animation in order to really fully climb in there. And I probably will get even more from it when I'm doing the drilling down myself. So I guess I'm just advocating for yes and more. I think that we need to meet people where they are. Some people are just going to want to play with the interactive thing, and that's all they want to do. Other people might really want to do the interrogation of the data. And, I guess relating it to my practice, and I think quite differently about data, but my ultimate goal, so I have these sort of images of artwork that are displayed on the website or videos, and they're static. And how does one actually interact with that if they're not in that exhibition? I think that's very difficult. I actually really think that it works best when people are building it themselves, or they go to the exhibition and they get to actually take home worms, you know, and then they're having a real relationship with the worms and interrogating the data like that. So that's my ultimate goal in the end, is to have somebody take worms home and have them and have a relationship. So that would be how, you know, I guess it's many scales is the answer.
Cool. Isabel, do you mind if I jump in for one of these questions? So
Go for it.
Shelby's asking, I mentioned that there are many people that are required to produce these images, and what does that mean? And I'll just say, so I'm one of the PIs, so principal investigators on this project, which typically means I get way more credit than I probably deserve for the sorts of things we're doing. There's a billion people that do this. I think one of them might be on today. I think I saw a message from Angie Heinrichs earlier.
Angie's here, yes.
Yeah, and she is kind of the, I think, the main person behind a lot of this stuff and how we use this in a web interface to help people interrogate their own data and other data. And of course, other people like Yatish Tarakia, who wrote the program that sort of makes the actual trees. It doesn't make them pretty, but it makes them. And then there's all sorts of other people that deserve tons of credit here too. So the Nextstrain team are people we've linked out to, right? They have this visualization platform. We did not build that. We just use it. And it's, I think the kind of broader point I want to drive to here is that the reason that we've been able to cover so much ground in a year as a scientific community is because all these tools are open access and freely accessible to us. So we know how they work. We can take them apart and we can link into those things as well. And I think that's been an incredible sort of team effort across the entire scientific discipline. And I've really enjoyed that.
So one of the things we've been learning through this genomics and art initiative is really how interlinked this idea of how interlinked these disciplines are and actually how valuable that link is. And so there's a question in here directly to Russ, but I think there's an element of this question that goes directly to Amy as well. And so for Russ, it's, do you have artists and designers working with you on these projects? And then I just want to frame it for Amy too. I think the corollary question is around the work that you do as scientists and what that adds to your efforts. And you talked a little bit about that in your presentation, and I'm wondering if we can elaborate on it.
Sure.
We start with you, Russ.
Yeah, sure. So the question of, do we work directly with artists? Not yet, but I'm interested in that. We have just started a conversation with our very first user experience expert, which is, I guess, not quite what you mean by artist, but certainly somebody whose job is to figure out how to make interpretation more straightforward. And that's, I think, a big part of what you probably do as an artist. And so we're going this direction increasingly because we know that to communicate this information, we need to figure out what will resonate simply and straightforward, which is something I've said many times, but remains true. So yes, I want to do more. No, I don't do enough yet with artists.
Yeah, I guess it's been fantastic getting to work with scientists. I do think sometimes I don't work with them for the reason that I think that oftentimes what they're really looking for is that specialist that Russ was talking about, this sort of information visualization specialist, somebody who can visualize this stuff very directly. And since that's not really what I do, I'm always like, well, you know, maybe I'm not the right person for that. But that being said, you know, there is this other realm where artists and scientists can collaborate that is a little more mushy and in between those things that can still yield really interesting projects that I think become accessible to the public as almost like a byproduct through their interactive qualities, or just making things visible in other communities that can happen through those collaborations. So it's been really great for me also because having somebody there to really explain the science to me and to help me understand that, who also then respects the art side and is curious about how I perceive what she's doing. And that conversation that we've developed over time has been really just super inspirational for my work. And I think probably for hers too, I'm guessing.
Thank you. I have this image in my mind from what you were speaking about just in the previous iteration, about us all coming home with a worm and exploring what happens when one does that. And so we have a question here about, and I think it's a really interesting question because it goes to the question of perspective and what it means, who you are or what you are that is seeing things. But also the social constructs around it and what data means. So here are the questions. So what would a non-human, such as a worm, consider to be data or work? And do they have social constructs like humans do? Amy, can you speak to that?
I really wish I could speak more authoritatively about that, but I can't. But I know that their data, they can sense light even without having eyes. Their data probably involves moist and dry gradients, where the best foods are in the spaces, which parts are easiest to eat. I think that they've got data that they're coming in through their skin and their sensory worlds. And I love trying to imagine what that's like. But I don't know what it is like. But I think it's really valuable to do that. I think it's a really great exercise to think about what things are like from the perspectives of worms and their symbiotic communities. I often wonder, what is their relationship like with the springtails that are there in the worm bin with them, and the mites? I think that they have an antagonistic relationship with the mites, probably a much nicer one with the springtails, just based on kind of what I observed.
And that idea of perspectives and what the data looks like is interesting for genomics too, because the question of what is the data that we're talking about and who understands it and what you see in the data is central to our effectiveness. And I'm wondering, Russ, if you can talk about the different kinds of data that you work directly with and how you explain it.
Okay. That's a tough question. So the short answer is there's a little bit of everything. Most of what we work with most directly would be a sequence. And so it's just a series of letters, basically. And there's actually only 4 of them. RNA and DNA are kind of a very simple alphabet. And so most of it is finding ways to use that kind of core low-level information to learn these other things, these sort of complex relationships among these samples, and to then lay on even more complex information like sample metadata, where it was collected, who did that work, that sort of thing, and try to build this all into sort of this complex net that weaves together all those little bits of data. So the short answer is there's a billion different kinds of data that we're dealing with. The longer answer is it's complex. It's a very complicated problem. That was sort of a way of not answering your question. I'm aware of that, but hopefully it answered part of it.
There's a kind of invisibility, I think, to genomics data for people that's interesting. So as Amy is looking at the worms and the plants and the structures that you feel, right? Russ is very much working with data that you don't see, right? It's computational data. And trying to explain that in a way that translates into something tangible, like what you're doing, Amy, is very difficult with that, with computing data, right? And so this issue of how we translate to the world is just central to what we do, because if we can't explain it, it isn't useful, and that's what we want to be, right? So. All right, we have one other question that I haven't covered, and this is directly for you, Amy. What are some examples of biomimicry projects focusing on mycelia systems?
That's a great question. I wish I could look it up and get back to you. I don't really know. I would love to hear if anybody did know about that. It's something I've been thinking about, actually. It's like, it actually seems like the internet is sort of a really good example of being inspired by fungal networks. Not that we necessarily got it from them, but it certainly mimics a lot of the same qualities of a distributed network that you could destroy parts of, and it doesn't destroy the whole network. The information can still travel. So that feels like a nice, at least, metaphor for a biomimicry project. In terms of actual physical projects, I have a book that I'm kind of excited about. It's under my computer right now, slime molds in arts and architecture. I feel like, thinking about computing with fungal networks is exciting. I don't know if we're quite there yet, but I love that people are experimenting with that. I'm not one of those people, but I feel like I want to know more about that personally. So I got the book, I haven't read it yet.
That's cool.
Jennifer, it looks like you just put a link in our chat related to this question, yes?
Yes. So Philip Ross, he actually was an artist in resident at UC Santa Cruz, probably close to when I first started working there, maybe like in 2003 or so. And he was an artist exploring, working with and making structures using mushrooms, and the ways in which you could sort of build these building blocks that could then become very sculptural, and making kind of these objects and environments, and has since gone on to really look at that more through the lens of sustainability and packaging, and has started a company and gone into this kind of innovative entrepreneurial way of being through this investigation that really started in the studio with these mushrooms that he was making, and building blocks from them. So Philip Ross, if you don't know his work, you can check it out.
Thank you. So there's this idea of complexity that's run through all of your work. Like, there's all these interconnections, right? We keep talking about that. And I'm wondering, one of the things that we think about all the time is how to represent that complexity, and yet at the same time simplify it. And so I'm wondering, and maybe Russ, we could start with you, if you can talk to what goes through your mind, particularly now in the new reality that you were describing of your new stakeholders that you work with, around how to structure the imagery so that you can show the complexity and the simplicity at once.
Sure. So usually when we're dreaming up a new way to look at data or to interpret these sorts of things, we just keep adding features. We just add new things to it until it becomes this enormous, extremely complicated, interconnected mass of stuff. And there certainly is beauty in complexity, but I think it's harder to get into that. And so the second phase of trying to turn this into something people can actually use and understand is to start stripping away all the stuff I added that actually isn't important, or is redundant with something else, or displays the same sort of thing, but really in a way that no one will ever understand, maybe including myself. And so I think it's sort of a process of getting to this complex thing, to just add everything in, the whole kitchen sink, everything I can think of goes into this project. And then we start saying, okay, but we got to cut that out, bring it back down into the simplest form that still has that maximum information content you're trying to get to. So it's sort of a complex process that gets us there, as far as I can tell, but it's working, and we're getting in closer and closer all the time, I think.
And Amy, a really similar question. Can you talk a little bit about how you integrate this idea of communicating the complexity of the networks and the processes that you're focusing on, while providing the simplicity that allows people to ingest that information and understand it?
That's a challenging question for me. I totally relate to how Russ is talking about it. Put everything in there and then strip it away. And I think that's the process of editing that artists go through as well. It's a similar thing where we do all of this research and we have all of these ideas. We at least me, I throw it all in there. And then when people go like, what? When I show it to people and they don't understand, I understand that I need to figure out a way to strip away elements and focus on the important parts. And then the other thing that I do is just work in series. So, I don't do one worm bin piece and try to throw everything into that. I sort of separate into several different projects that each focus on one element. So, I can think about worms in relation to eating in one project, worms in relation to sound in another, worms in relation to humans moving in the rocking chair and how we're participating in a different project. So they don't all become one confusing mass together. It's kind of how I think about it.
Thank you. I wanted to give a moment to Jennifer Parker because she has conceptualized this initiative to talk about how we bring these ideas together, both in terms of thinking about it now, but also what are our next steps? How do we continue to think about it and how do we evolve?
For me, they've never been separate. It's sort of this idea of being an observer in the world. And I think artists and scientists look through all kinds of lenses, whether it's through, the telescope or the computer, which is a kind of lens, or our eyeballs, or, the magnifying glass and the microscope. There's all these ways in which I think we come together to really ask questions and have curiosity. There's nothing new here, and we've really been good at sort of unsymbioting ourselves, de-siloing or siloing ourselves into these tiny things, and taking apart the ways in which information has fed itself through these sort of organisms of humans that we've now sort of put in boxes and kept ourselves in the research institute as separate and disciplines as separate, so much so that our students are like, oh, this is my major, as if they didn't really belong to a larger system. And I think part of, creating Open Lab, the collaborative research center that I run, and engaging with faculty and students across campus, but then opening that up to the general public and other artists and scientists across the country and in different institutions, is to sort of remind ourselves that these are artificial walls that we've put up, and how we start to come together and think differently and open up and expand ways that we have looked at these questions and ways of considering the world for a very long time prior to progress in the last 100 years, when we really started narrowing our focus of disciplines and creating these sort of monoliths of departments and everything. So in some ways, it's just kind of peeling back that onion skin and going back to sort of the inside of it and saying, oh yeah, these are things that we share in common, and being able to feel comfortable and open with each other, to ask questions, to go to your colleagues in departments that you're not part of. So it's kind of silly that I've spent so much of my career sort of building bridges, but I think we're all here now, and it's not, I don't think we have to talk about art and science anymore. I think we can actually talk about curiosity and wonder and figuring out, more ways of collaborating that also support the disciplines across sectors. So I'm just excited to continue to work with Genomics Institute and to have Amy here and some of my other colleagues and people that I know from Ohio State, and feeling like, in our complete exhaustion and the gift that you guys gave to sort of be articulate in your practice in this 1 hour, that's something that we didn't do even, last year and a half. so that to me feels great. And I'm just, pleased to see where we go from here and how we can sort of not have a short-term memory loss with COVID if once things really open up again, and take these practices into consideration into the future. So I don't know, I'm not trying to wrap it up in any big picture because it's very complex. But, I also invite more people who are curious to want to work with the Genomics Institute to contact me through Open Lab. You can find me in the art department at UCSC. Obviously, I think there's a lot of curiosity to want to work together, and, for Amy to want to continue to work with us in any way that she's interested.
Absolutely. And thank you. I mean, and Jennifer, you And Isabel, through the pandemic, we were able to put this art and genomics convergence together. And we mentioned earlier, we wouldn't have been able to do it in the physical world because it's just too complex and too big and doesn't fit in our little gallery. So we're excited that this is a launch and it will continue, and we will do more programming like this and open up to as many collaborators as possible. So thank you. I want to just show a slide of what's up next at the Sesnon Speak Up, and stay involved. Coming up on Monday, we're having our Sesnon Speak Up of Revolutionary Greetings, which is part of the Solitary Garden and part of the Barring Freedom exhibition that is about to close at the San Jose Museum of Art. And the Solitary Garden works with Tim Young at San Quentin. And then the next event next week is the First Generation Arts Alumni Virtual Exhibition. We are reaching out to all first gen students and talking about their experiences at UC Santa Cruz and how their career path has evolved since that, and that's exciting. And we'll do a Sesnon Speak Up called Visibilizing Possibilities, which means to make visible things that have been erased. Or so anyway, that's a new concept, but it's not unlike those worms underground, making things visible again. So I just thank you all and welcome you to collaborate with us and keep us posted on all of these fantastic projects. Thank you.